All right, guys, so in this video, we're going to be talking about biotech and specifically bacteria transformation. So there's a lot of uses for this in terms of why you should really care about it. But basically, we can do all kinds of crazy things in terms of getting a small sample of DNA and amplifying that amount by PCR. We can actually cut and insert genes. We can actually select genotypes and actually breed them in a lab setting. So all kinds of crazy stuff that we can do with biotech. So just to kind of put this into context, right? We'll introduce this in terms of like diabetes, right? So basically the pancreas is going to secrete insulin as a nice little review for cell signaling. Insulin binds here to a receptor protein that's allowing glucose to go in, and then glucose can actually be broken down. Now, if you are type 1 diabetic, the pancreas actually cannot produce insulin. So that way your glucose and your blood sugar levels get really, really high. And you can't actually, oftentimes these people are super, super lethargic because they can't actually break it down, and then it passes through. So it's a lot more problematic. And the treatment for this would be that they would have to actually get this manufactured insulin. Now, if you're type 2 diabetic, your pancreas works fine in the sense that you can still make insulin, but your insulin receptors are no longer functioning, or they become desensitized to insulin. And you run into similar issues. Your blood sugar levels get super, super high, and the cells are going to fail to respond to that insulin. Now, I'm going to specifically just mention why we should care about this in terms of type 1 diabetics, because as you can see here, that this pancreas secretes a protein, right? Actually, I should show you here, right? This protein is secreted via exocytosis, right? So via the endomembrane system, right? So try to kind of put all these pieces together, right? If this is a healthy pancreas, all of these cells are doing protein synthesis, and they're producing a protein that's shipped out through the bloodstream, and that signal is trying to find the right receptor protein to actually let glucose in. So there's all kinds of concepts that we could throw in here, but let's kind of talk about, if you are type 1, where do you get that insulin from? That really comes from genetic engineering. And so basically this is going to allow us in a lab to actually manufacture proteins. Now, when we say we're manufacturing proteins, the proteins are still very much legit. They're going to be the same human proteins that you would need, but insulin is going to be produced via this genetic engineering. So we take the gene from a human, we put that gene into a prokaryotic cell, and that's going to make that protein for us, and we can harvest it later. So oftentimes, in terms of what we should really focus on in terms of this unit, is bacteria use those really tiny circular plasmids, right? So if I drew just, I don't think I have a picture here, but yeah, so let's talk about this plasmid. So if you don't know, remember E. coli, and go back and watch my videos on this in chapter 18. E. coli has this really large circular chromosome, but also too, there's really small circular DNA called plasmids. Now this is not to scale, but there's a lot large. A bacteria cell can have quite a few number of plasmids. And so basically what we're doing is we're going to take the human gene, put it into a plasmid, and insert that plasmid into bacteria. And so bacteria uses those plasmids. Now, when we isolate a plasmid, we call it a vector. So basically, if I were to draw a simple picture over here, if this is my prokaryotic cell, there's the main DNA. What I'm trying to do is take this, what's called a vector, which a vector is the same thing as a plasmid, it's just isolated. And I'm trying to get that DNA into the bacteria, and that way that bacteria can actually express that gene. So basically what we do, and if I were to kind of draw this out, let me try to color coordinate this. Let's say right here, if this is my vector, and I made this in a lab setting, and it's in purple if you can't see. Basically, let's say that this thing in purple right here is my gene of interest. So basically, when we take the plasmid with our gene of interest, let's say that this would be the gene of human DNA, right? Human DNA of insulin. If I isolated that gene from a human, I made a plasmid in a lab, and I put that plasmid into the bacteria, well, this bacteria is going to just do protein synthesis and make that insulin. So it's really, really cool in terms of what we can do here. So a couple of words to remind you guys. Anytime you manipulate genes or you get a new type of DNA, we're going to call that recombinant DNA. So you get DNA from 2 or more sources. So And it's pretty interesting. And the big thing that you also need to remember is once the bacteria has that plasmid, as it replicates, it's also going to replicate that plasmid. So it's going to get passed down again and again and again. So basically, like I said, you're going to take that human DNA, put it into a plasmid, and that is going to be your whole genetic engineering side of things. So let me show you, I want to say I have a visual pulled up here. So in terms of this one, actually, I'm going to hold off on that little quick clip. But before I do that, some of you guys might be asking, how do you cut out those enzymes? Or how do you, not the enzymes, how do you cut out the DNA fragments to make a plasmid? Well, that's where we run into things called restriction enzymes, okay? And so what restriction enzymes do is, depending on what the enzymes are, they're going to cut the DNA at different sites. So this is where I wanted to show you this little visual. If I wanted to actually show you this, these restriction enzymes, if I clicked on one of these, and it recognizes that what are called palindromic sequences. So that means as it's read in one direction, it's going to be read the same in the reverse. So GAATTC. In this direction, it's going to be read the same at GAATTC. So these enzymes are going to search the entire genome for that one particular site. Another enzyme is going to cut here, another enzyme is going to cut here. So if you just watch this, the restriction enzyme is going to actually search and find this sequence, and it literally cuts the DNA at that site, and then it breaks it up, and then we can actually add it in together. Okay. Now, how do we actually, and why do we care about that? Well, remember, once we kind of figure out where the gene is, we can cut open the DNA from a plasmid, and then we're going to use DNA ligase to seal it back together. So we can insert our human gene of interest, and then we're going to basically seal it using DNA ligase, the same enzyme when your DNA seals those Okazaki fragments in DNA replication. So it's pretty interesting when you think about everything that's going to happen when just making a plasmid. So in terms of that, let's kind of jump back to this, and I want to make sure that I'm not missing any visuals I wanted to show you guys. Yeah, so this is basically showing you what's going to happen here. We're going to try and try to put this together now. Here's your restriction enzyme, right? Right here, there's one of those sites that it's actually going to cut it up, right? So it's going to cut the sequence out, this restriction enzyme site. And then all of a sudden now I've opened up my plasmid. This is, again, going into my bacteria. I'm going to use that same restriction enzyme or a different one, doesn't matter. And I get my human gene of interest, and then I'm going to use DNA ligase to seal this back up, all right? So basically that's kind of insane. We can actually cut a gene out, put it into a plasmid, and then basically what we're going to do is we're going to seal it up together using DNA ligase, okay? So like I mentioned, as it seals, here's your plasmid. This thing in black is going to be your gene of interest, okay? Now, we can manipulate this in a number of different ways, but it's pretty interesting, and I just want to show you this quick little visual. Like I said, if we get bacteria to have that gene of interest, we're growing it on a petri dish of E. coli, we're going to let this thing grow, and it's going to replicate not only the bacteria itself, but it replicates that plasmid that we added as well. So that's something that I think is really, really interesting. And again, the key thing is once it has this gene, it's going to make that protein. So it allows them to actually survive and reproduce and make that protein that we wanted it to do. So that's pretty interesting. So that's what those restriction enzymes are, and that's really what their focus is. But let's kind of get into what our really big fixation is on this bacteria transformation line. Just kind of reminding you, this is showing you the bacterial genome, but let's talk about transformation in one little lab that might make sense. Basically, this is showing you over here on the right-hand side is that these mice, basically they injected one rat with a deadly bacterium, and then obviously the mice died. But they also injected the mice with a dead version of the bacteria, and the mice still died. Now, the reason why that happened is because of what's called a transformation. Now, go back and watch my video on chapter 18, but remember, transformation is when bacteria cells can actually share plasmids from one to the next. So that's something that I wanted to kind of revisit this. So bacteria can actually share plasmids with one another. So that's kind of interesting. So like I mentioned, these bacteria cells, they have those little tiny plasmids. This is a much more appropriate version of drawn to scale, where there's a large circular chromosome, has all kinds of genes that the bacteria would need to express to actually just survive. but also these other plasmids have other genetic information, okay? So how can these plasmids help us? Well, kind of like I just mentioned, right? We can manipulate it, we make a vector, we can inject that bacteria into, we can inject that plasmid into the bacteria and it's gonna express it. So let's kind of move on. We talked about restriction enzymes, like I mentioned again. And I do want to mention this again. The code is universal. When I talk about the code, I'm talking about the genetic code, meaning UUA. Let's say if that's your, your RNA codon, it's going to code for leucine. Doesn't matter what kind of cell it is. Same thing. If I had ACU, that's going to code for this amino acid threonine, right? If you don't remember this, go back to the videos in chapter 17 on protein synthesis. But basically, everything is going to read their genes the same way. The coding sequence is going to be the same. So that's why it's really, really beneficial for us to actually use these bacteria cells. So let's kind of get into a little more of the specifics of what we really should need to know, okay? The lab that we did is we used the glowing gene. Actually, I think this year we had that red zebrafish gene that was actually used instead, which is really cool. But basically, when we talk about transformation, like in a lab setting, right, we're going to take one of these plasmids, it's going to uptake that foreign DNA. So this whole thing is your bacteria cell, and we're trying to get that plasmid in here so that bacteria can actually express it, okay? Now, what is our plasmid really that we are going to be fixated on is going to be a few things here. So let's talk about the plasmid that is typically going to be used again and again and again and again, okay? Now, here's your pGlow gene. Now, most of these are going to be working very, very similar. Let's talk about it. So the ORI just stands for the origin of replication. I'm not going to talk a whole lot about that. But let's kind of talk about the way that I like to think of these operons. Now, this GFP, right? Now, this can be anything, but basically how I want to think of this is the gene that you want expressed. Basically, this is the one that's going to make it, in this example, this is the gene that was going to make those bacteria cells actually glow. Okay, so this is the gene that you want expressed. In this case, we're using the glowing fluorescent protein, but all these operons are going to be manufactured mostly the same way. So the other thing that we have is going to be the RSC. Now, this RSC is actually going to be an operon for the GFP gene. Now, I'll put in parentheses, or your gene of interest. So basically what I'm meaning that is that this GFP can be anything. In this example, it's that jellyfish gene, but whatever we wanted, if this was insulin, it would go here. But basically, always before this GFP gene or your gene of interest, you're going to have an operon, okay? So it's an operon, and the way I think of it is basically this turns on / off your GFP. Okay, so this is going to turn on or off that GFP gene. All right. Now let's talk about one of the greatest names in terms of gene naming, which is going to be the BLA gene. And basically, this gene makes all bacteria resistant to ampicillin. So basically, this BLA gene is going to make it resistant to ampicillin. So you might be asking yourself, well, why do you really care about that BLA gene? Why do we add this? And that's a great question. So ampicillin, if you don't know, is basically a gene. Well, I should say ampicillin normally kills bacteria. Now, we're basically adding that BLA gene to make bacteria survive in the presence of ampicillin, okay? So the question really should be, why do we add this? And the reason being is because we always stick that resistance to ampicillin in these transformation labs, because if it can grow in ampicillin, so if the bacteria grows in ampicillin, that means it 100% confirms the plasmid was received. So basically, we got three genes here. We got the RSC, which is going to be an operon for this gene to be expressed. This one is going to control the on or off if you're going to make that gene. We also inserted the BLA gene that makes it resistant to ampicillin. The reason why we cared about that is because we grow bacteria in ampicillin when we do transformation, because if it survives in ampicillin, the only ones that survive are the ones that have successfully uptaken or received that plasmid. So that's kind of the ins and outs. So let me kind of get this dialed in, and a lot of this just basically is another thing, another visual of this. So we're basically going to use it. Now, let me kind of double check to make sure I'm not missing anything here. So basically, the bacterium, if we looked back here, if this Petri dish was coated with ampicillin, and I get some bacterial growth here, these are not just like one or two cells. These are actually a couple of hundreds of thousands, if not, of little bacteria colonies. Now, if this whole dish was covered in ampicillin, these ones that survive are going to be the ones that are going to be resistant to ampicillin. So you know for sure that they've received that plasmid. So let me just double check to make sure I'm not missing anything on the analyzing the biotech side of things on the transformation lab. I don't think I am here, but let me just kind of introduce this and kind of show you a typical kind of response here. So the most confusing thing is, at least for a lot of students in my experience, is we know the plasmid, right? We got the BLA, the GFP, the RSC. The thing I want to take a second and just remind you guys is what's up with this RSC, okay? How does this actually control the GFP gene? The RSC controls the expression of GFP, okay? Anyway, let's get this dialed in. So the goal, like I said, is we're trying to get it to express that glowing gene, but here's kind of the RSC, okay? Now, let me try to draw this out as simple as possible and try to make as much sense of this to make it simple. So the way I think of it and the way that I want you guys to make sure you guys feel comfortable with is that if this is my plasmid, right here is a zoomed up version. This right here is a zoomed up version of that RSC. And then right after that is going to be your GFP gene, right? So down here, I would have my GFP gene or my gene of interest, right? So basically what this looks like, all you really need to know is that arabinose turns on the expression of the GFP, okay? Or your gene of interest, whatever that gene of interest might be, right? So this would be your GFP, okay? So let's kind of get this dialed in now and try to make as much sense as possible. So basically arabinose, if you look here, you have an RSC, this is your gene here, right? It's an operon, and arabinose actually is gonna be an inducer. It's an inducible operon. So you might want to go back and watch the video on inducible operons if you're feeling confused on this. But arabinose binds to that gene, and it basically changes the repressor protein shape and allows it to be expressed. Okay, these genes will now be expressed. So simply put, you just need to know without that inducer, the inducer in this case is that arabinose sugar. When that sugar is there, the GFP gene gets expressed. Without the sugar, you can see RNA polymerase won't be able to fit and it doesn't express the genes, okay? So let me kind of give you a rundown of kind of how these experiments would look. And what we did in the lab, well, and if we're watching this and we haven't done the lab yet, this is typically what we're gonna be looking at, right? So basically we're gonna have three dishes. Each one of these are gonna be Petri dishes. So I'm gonna call this dish number 1. This would be dish number 2. And this would be dish number 3, okay? So I'm gonna review a couple of things. P with the minus sign next to it means the bacteria without the plasmid added. P with the plus. P plus is going to be the bacteria with the plasmid. All right. Now, there's a few other things too that we should be aware of. Then I'll kind of color coordinate this as best as I can. So I'm going to talk about Lorry-Broth. Okay. So I'll just abbreviate it Lorry-Broth, which is, I'm just going to abbreviate LB, is food for bacteria. Let me switch up the colors here. I'm going to do, let me see here. I'll do blue for my next one. I can move this one out of the way. So I will do blue is going to be ampicillin. And like I said, that's typically going to kill bacteria. And let's pick one final one. The purple will be arabinose. And that's going to be the sugar that turns on the GFP gene. All right. So let's talk about how this experiment typically is going to be run. So I have dish one, and on dish one, I'm just going to put Lorry-Broth only. Okay. I'm going to put on dish two, I'm going to add Lorry-Broth. Dish three, I'm also going to add Lorry-Broth. This second dish is going to get a treatment of ampicillin. So I'm going to put Lorry-Broth plus ampicillin. Okay. And I'm also going to add ampicillin to this last one. And this last one is where on dish three is where I'm going to be adding in my arabinose. Okay. So hopefully you're sticking with me and you kind of get an idea of what's happening here. On this dish, I have just Lorry-Broth. Okay. So that's the medium I'm going to be growing my bacteria in. Dish number two, I have Lorry-Broth, but I also had ampicillin covering the entire dish. Okay. The third dish, I have Lorry-Broth, I have ampicillin, and I also added that sugar. Okay. So basically what we would do in a lab is on one side, we added bacteria without the plasmid. So I'm going to just say P with a minus sign next to it. So basically I took all of my bacteria without the plasmid as my control group and I added it here. On this side of the Petri dish, I added all my bacteria with the plasmid. Okay. And I repeated this for all three. So I have the P with the minus sign, P with the plasmid, P with the minus, P with the plasmid added. Okay. So on the left side, I added bacteria without that plasmid, and the plasmid I'm talking about is this plasmid, right? So go back and re-listen to the explanation about each gene, but let's kind of clarify what's happening here. Now, we're going to make some predictions of what's going to happen. Let's take a look here and see the growth, okay? So in this first dish, all I have is food, just food. So basically what's going to happen is over time, I would get complete growth, and all of this stuff I'm drawing in is going to be bacteria. So basically it's really gross. But the reason why I have complete growth is there was nothing there to kill off the bacteria, okay? So I'm just going to put this here, complete growth. Now, the reason being is nothing's there to kill it. So common question would be, do you know which bacteria had the plasmid on this side? You don't. And that's the reality of it, is because there's nothing to kill off bacteria. Doesn't matter what bacteria I had, I'm just going to get complete growth. Now, in the second dish, though, is where I added ampicillin. So ampicillin kills bacteria. So if I looked at the bacteria that did not get this plasmid, I didn't add that plasmid to it, all of my bacteria is going to die. So I'm going to get no growth on this. But on the plasmid side, I'm going to get some little tiny colonies. And it might look something like this, okay? And basically, the reason why I did it this way is because, okay, I get partial growth. And the reason why I just got partial growth is because only a small percentage receive the plasmid. And that's the reality. Only a small percentage will actually pick up that plasmid. But the good news is that the fact that these ones actually still survived when this whole dish was treated with ampicillin, these ones that survived, for sure, you can confirm actually had received the plasmid because they can survive an ampicillin. Now this last dish, let's look at the difference between them. And I'm using it with the example of that GFP. Basically this one, I'm gonna get partial growth on the P+ side because I have 3 things now added to this dish. I have Loribroch, ampicillin, and arabidose. Now, this one is the one that actually gets that gene, that green protein expressed. Now, the reason why I actually had that green protein expressed is because I had arabidose. And remember, arabidose is basically that inducer, like I mentioned a second ago, that turns on the expression of that GFP gene. So what's the difference between dish 2 and dish 3? Well, I have no growth on the P- side because that bacteria didn't receive the plasmid, so it killed off all the bacteria because they don't have that resistance to ampicillin. I have kind of a similar percentage about the bacteria that's actually expressing that green, or I should say surviving on the P+ side, but the difference is that that P+ on the third dish, there's that arabidose, okay? And arabidose is the inducer to actually turn on the expression of that gene. So basically this experiment is really doing a couple of things. It's talking about transformation, but it's also about gene expression in prokaryotes. So that is the basics of bacteria transformation. I want to say I do have a sample question pulled up, actually. I'm going to call it there. And we will see you guys in the next video.